Okay, this is the third lecture on water and wastewater treatment section. In previous lecture, you learn about water chemistry and you learn about the constituents of wastewater. There are three types: biological constituents, chemical constituents, and physical constituents. Under the biological constituents, you learn about fecal coliform or coliform organisms, which are the pathogenic microorganisms can be available in wastewater. Then under the chemical characteristics, you learn about pH, BOD, COD. Now all of you know the meaning of BOD and the COD. Okay? So today lesson is again about the BOD and also about the physical characteristics. What is BOD? BOD represents the amount of organic matter available in the water which can be degraded biologically, or the simple meaning of biochemical oxygen demand is the amount of oxygen required to degrade the organic matter biologically. What is COD? COD is the chemical oxygen demand. So it is about the demand. It is about oxygen demand. That means oxygen is required for some purpose. Okay? So purpose is to degrade the organic matter into simple end products. So amount of oxygen required to degrade the organic matter chemically is the COD. Then usually BOD is smaller than COD value. Okay? You may have checked it with the tolerance limits also. So tolerance limits of BOD for industrial waste into inland surface water is 30 milligram per liter, while tolerance limit of COD for the same case is 250. What is the reason for that? All of you know that by using a strong oxidizer or chemical, we can degrade all the matters available in the wastewater other than using biological method, using microorganisms. Then there's a BOD5. Okay? So in tolerance limits standards, you have seen that BOD5 is measured at 20 Celsius and BOD3 is measured at 27 Celsius. So what is the reason for 5? 5 means within 5 days. The amount of oxygen required to degrade the organic matter within the 5 days. BOD3 means within 3 days. But there is a reason for that. So you may think that reason is something ridiculous or humorous, because this standard BOD test was first used in UK. UK scientists found that the longest river in the UK took 5 days to flow from the river source to the sea. Okay? The longest river took 5 days to flow from its source to sea. That means when one water drop generated from the generating point, it took about 5 days to reach the sea. So it is ridiculous, but it is the reason for taking BOD5. What is the purpose of using BOD? BOD means amount of oxygen required to degrade the organic matter biologically. Let's see what are the methods, what are the situations that we use BOD. So to determine the quantity of oxygen that will be required to biologically stabilize the organic matter. So if we know the BOD of the wastewater, then according to the tolerance limits or standards, National Environment Act, NEA, according to the NEA Schedule 1, 2, 3, we can find the tolerance limits of the BOD for whatever waste to discharge into whatever place. Then if we know the influent BOD, then we can calculate the quantity of oxygen that will be required to biologically stabilize the organic matter. So there are some processes where we have to supply air or oxygen for the process. Then again, if we know the BOD, we can determine the size of the wastewater treatment facilities, size, capacity of the tanks. Okay? In a wastewater treatment plant, there are many tanks, there are many unit processes, there are many unit operations. Then we can calculate the capacity or the size, and we can determine the required facilities, whether we need a aerobic tank or anaerobic tank, like that. And also to determine the efficiency of some treatment processes, we use BOD. So consider this is a sedimentation tank. Okay, so this is the inlet BOD. I'll use BOD. Okay. Inlet BOD is BODI, and outlet water BOD is BOD E, effluent, influent, effluent. Then we can remove sludge from the sedimentation tank. So we can calculate the efficiency by knowing the BOD difference of both here in influent and here effluent. BOD difference by the inlet BOD. Okay. So if we know the efficiency, if we know the BOD, we can calculate the efficiency of the process. So we can compare the wastewater treatment plants. We can compare this method is good because here there's a 50% BOD reduction. Like that, we can compare. And we can determine the compliance with wastewater discharge permits, because industry has to comply with the NEA standards, National Environment Standards. They have to follow the NEA. So if the BOD value has matched with the NEA standards value, then no need to worry about the process. So what are these? So when you do the practical, you will be familiar with this. These are BOD bottles. So when we use these bottles, when we check the BOD of the water sample, so these are digitally facilitated BOD bottles. So these are the conventional BOD bottles that we use to do the practical. So this is usually BOD bottles are in brown color or black color. So normally we do not use transparent color, transparent BOD, transparent bottle. So here it is connected to a YouTube manometer. So whatever manometer. So usually these manometers consist of mercury, because you know the mercury has a higher density. So because of that, we can reduce the height of the uh, YouTube manometer. If we use alcohol or water, then what happen? Height of the manometer will be increased because water and alcohol density is lower than that of the mercury. When you do the practical, you will understand what is the purpose of having manometer here, because in this method is used called as manometric method, which is for BOD measurement. So you will understand what happened in the BOD test later. So this is a BOD bottle with digital system. So here the pressure sensors are available. The pressure sensors are available, and digitally everything is converted into uh, to the reading. Okay, so here you can find BOD is a chemical procedure for determining the amount of dissolved oxygen needed by aerobic biological organisms in a body of water to break down organic matter present in a given water sample at certain temperature over a specific period. So in our previous lecture, you learn about these things, and it is not a quantitative procedure. I'll see. Um Here we'll see here. Okay, it is not a quantitative test, although it is widely used an indication of the organic quality of the water. Okay, remember it is not a precise quantitative test because some errors can be occurred, but it is always used in industries. It is most commonly expressed in milligrams of oxygen consumed per liter. Milligram per liter. The BOD unit is milligram per liter of sample during 5 days of incubation and 20 Celsius. So BOD 5 is taken at 20 Celsius. It's often used as a robust surrogate of the degree of organic pollution of water. That means we have to keep the wastewater samples at 20 Celsius temperature. That is why it is called as incubation. Then uh, as in our previous slide, we can see BOD can be used as a gauge of the effectiveness of wastewater treatment plant. To measure the efficiency of the plant, we can use. Then here, as in previous slide, we can see here the 5 days. 5 days was chosen as an appropriate test period because this is supposedly the longest time that river water takes to travel from source to estuary in the UK. So this is something ridiculous, but it is the reason. So it is the reason for taking 5 days, but now it is internationally recognized standard. In the presence of pre-oxygen, aerobic bacteria use the organic matter in wastewater as food. So you may think, what is the in our previous lecture I also explained that in the presence of pre-oxygen, aerobic bacteria use the organic matter in wastewater as food. Or aerobic bacteria eat organic matter. This is the simple meaning. BOD test is an estimate of the food available in the sample. All of you know BOD means amount of water, amount of oxygen required to degrade the organic matter biologically. So amount of oxygen required to degrade the organic matter. So who degrade? The microorganism degrade the organic matter into simple end products. So microorganisms eat the organic matter. So BOD test is an estimate of the food available in the sample. The more food present in the waste, more dissolved oxygen will be required. So if there are more food mean organic matter. Organic matter, all of you know organic matter are uh, carbon, hydrogen, nitrogen, oxygen, there may be sulfur also. So if there are more organic matter, then more dissolved oxygen is required. Therefore, if there are high BOD mean high organic matter. BOD test measures the strength of the wastewater by measuring the amount of oxygen used by the bacteria as they stabilize the organic matter under controlled condition of time and temperature. So, how BOD different than dissolved oxygen? Remember, BOD isn't really there. So, if we talk, always we know pH is an indication of H plus ion. POH is indication of hydroxyl ion. So, likewise, BOD is not available. It is an indication of amount of organic matter uh, in organic matter that can be degraded biologically. Okay. So BOD is not really available. It is an indication of dissolved oxygen. BOD is the amount of oxygen that will be consumed given sufficient time bacteria oxygen to completely decompose the organic matter. So this is the reaction. These are the organic matter: carbon, hydrogen, oxygen, nitrogen. Here sulfur is not available, no need to worry about that. So oxygen is required, then carbon dioxide, H2O. If there are nitrogen, they will be converted into ammonia. If there are sulfur, they are converted into sulfate. So this is the balance equation. If there are in amount of carbon, then they will be converted into NCO2. If there are a amount of H, then this is the H2O. Okay, so no need to hang up on the numbers, and no need to remember all these things. Just uh, remember these things. Okay, no need to remember the balance equation. Then in our laboratory class, we have to determine the BOD of a wastewater sample. So in my lecture, I'm not going to detail about the experimental procedure, but I'll just give some hints. So how would you determine the BOD? First, we have to take wastewater sample. Okay, uh, we should have a wastewater sample. Then we have to add bacteria. Sometimes there may be bacteria in the wastewater. Then we do not want to add. So how can we find bacteria? We can ask some bacteria from a wastewater treatment plant. We can take some sludge as the bacteria, because in the sludge bacteria is available. Then we have to add oxygen quantitatively. Wait until all the organic material is gone. So we have to provide oxygen. So no need to give oxygen. If there is enough space in the bottle, then oxygen is available. Then there should be a proper stirring method or mixing method. Then after several days, we can measure the amount of dissolved oxygen available in the water. So here now you can see this is time versus BOD remaining, or BOD remaining, or L. Okay, so L is the BOD value. Okay, so here you can see L or BOD remaining. So here at the beginning, the BOD of the bottle, you can see BOD remaining is very high. That means at the beginning, remaining organic matter amount is very high. At the end, that means we have to take a end time period. So at the end of the time period, the organic matter content is very low. That's why BOD remaining is low. What is BOD remaining and the BOD T? BOD remaining means the amount of organic matter uh, remain in the wastewater sample that can be degraded biologically. But BOD value is the amount of oxygen required to degrade the organic matter biologically. So here now you can see this is BOD extended. That means the BOD, the correct BOD value. So with the time, BOD value is increasing. With the time, BOD value is increasing for a particular time period, but After that particular time period, BOD value not change. It is remain uniform. The reason is BOD value is not changed, it is uniform. That means there is no addition of, there is no consumption of oxygen to degrade the organic matter. It may be due to the complete exhaustion of organic matter. So and BOD, this is BOD remaining. What happened until particular time period, BOD remaining value is decreasing. Okay, this is BOD remaining until a particular time period. The BOD remaining value is decreases, and when it reaches to that time period, that particular time period, and beyond that particular time period, BOD value will not change. It is kept uniform. Okay, because consider that we take a wastewater sample, and we with the time period we check the dissolved oxygen level using a dissolved oxygen meter. So first that means we measure dissolved oxygen level of the water, mean we measure the BOD remaining. Okay. First we identify dissolved oxygen level is high. With the time it is decreasing to a certain value. Then what happened? Now we have to assume that there are excess amount of dissolved oxygen in the wastewater sample. So there are what happened? This dissolved oxygen is consumed by microorganism to degrade the microorganism biologically to do this reaction. Then after completing degradation process, there is nothing to do with the dissolved oxygen. Therefore, dissolved oxygen level remain constant with the time. So when you do the BOD test, there are some limitations due to the availability of sulfur and nitrogen substances. Only 60 to 70% of soluble organic matter is metabolized in 5 days. Inorganic oxidation may also occur. Then if there are sulfur, inorganic oxidation of sulfur or iron, if there are sulfur or iron, what happens? Sulfur will consume oxygen and produce sulfate. If there are ferrous, they will consume oxygen and make ferric. And nitrification also can occur. Nitrification may also occur if ammonia is present. So if there are ammonia, then they are converted into nitrate. So to this, to convert this reaction, oxygen is consumed. Actually, these these are not actually organic matter. So organic matter consists of CHO, and there may be nitrogen and sulfur in the organic compound. But if there are inorganic, sulfur and nitrogen, what happen? They also consume oxygen. So then our the measurement, the reading of BOD value can be BOD value is wrong. Therefore, there should be a method. So in our practical, we add nitrification inhibitor for that. Then this is about details of nitrification. Then in your first year knowledge, or in your A-level knowledge, in your semester 1 knowledge for the chemical students, or from your A-level knowledge, you can remind about the reaction kinetics. Okay, BOD reactions represent first order kinetic. Okay, first order kinetics refers to the dependence on the concentration. So the rate of the reaction What is the reaction is organic matter are converted into CO2, H2O and other simple end products by consuming oxygen and the, by microorganism. So rate of reaction is the change of reactants by change of time. So it is equal, the rate of reaction is equal to the reactants, K by reactants. Okay, K is called the rate constant. It is only that thing that is constant in the kinetic stream. So the rate constant depends on the reaction, type of the reaction, depends on the type of bacteria. So if it is a BOD, and depends on the temperature. So here, now you know about BOD reactions, BOD variation with the time. Here, this is time versus BOD curve. So this is BOD T value or the BOD exerted value, the correct BOD. And this line and this blue color line refers to the BOD remaining. Okay, so then what is this value? This is ultimate BOD. At the end of the process, the BOD value comes to a certain value. It is called at ultimate BOD. Or when the time is at infinity, the BOD is BOD value equals to ultimate BOD. Okay, so this is the ultimate BOD. So according to this figure, ultimate BOD value is around 120 milligram per liter. So ultimate BOD, if you determine the BOD after 5 days, this is called the 5-day BOD. So here this is day 5. So at day 5 it is around 100, and at day 20 it is 120. I think these terms are not, this is wrong. Okay. So if you determine the BOD after 20 days, this is called BOD 20. It is correct. Okay. So here I have to remove this. Okay. Okay, then now it is correct. If you determine the BOD after 5 days, it is called BOD 5. If you determine BOD after 20 days, it is BOD 20. These are really BOD exerted values or BOD T. We can use BOD T. The ultimate BOD is the amount of oxygen required to decompose all the organic matter after infinite time. So what is that? Infinite time. According to this figure, that infinite time is 20 days, because after a certain period here BOD values kept constant. So it is 120. So after 15 days, okay, so we can assume B15, BOD 15 and BOD 20 is 120, but BOD 14 is not 120. So here ultimate BOD value is 120. Okay. So this is usually simply calculated from the 5-day data. Because we cannot wait for the infinity. Most of the industries, they take ultimate BOD as the 5-day. They assume within 5 days all the and many parts, okay, many part of the BOD organic matters are degraded. Now, according to the kinetics knowledge, now we can write the rate of the reaction equal reactants, change in reactants, change in time. So change in reactants are the BOD remaining. Reactants are the organic matter. So remaining organic matters are the reactants, and dt, then we can write it is as minus K1 BOD remaining. T is the time, BODR is the amount of BOD remaining at time T. K1 is the first order reaction rate. Usually it has units 1 day, 1 per day. Then we have to integrate the above expression between the limits of the ultimate BOD and the BOD T, and we can derive this equation. Then we know this one, BODT equal ultimate BOD minus BOD remaining. Then by using the first expression, then we can derive an expression for the exerted or BODT as this way. Okay, so this is the expression that we can use for many calculation. If we know the ultimate BOD and we should know the rate constant, then we can calculate the BOD at time t. Then here you can do this simple exercise. It is given BOD 520 milligram per liter. It is given temperature 20 Celsius, K1, and we have to find BOD2. You can try using those equations. Then COD. The basis for the COD test is that nearly all organic compounds can be fully oxidized to carbon dioxide with strong oxidizing agent. Oxidizing agent is the dichromate, okay, under acidic condition. So usually this is done at acidic condition. The amount of oxygen required to oxidize an organic compound to carbon dioxide, ammonia, and water is given by this equation. And you know, COD value is greater than BOD, or BOD value is smaller than COD. So it is because many organic substances which are difficult to oxidize biologically, such as lignin, can be oxidized chemically, because some chemicals can be easily degraded by chemicals, dichromate. Then inorganic substances that are oxidized by the dichromate increases the COD amount of the sample. Presence of inorganic substances with which dichromate can react. There are some inorganic substances. Because of that, inorganic substances also react with dichromate and consume oxygen. Because of that, the COD value can be increased. Then when we talk about the biological degradation, some organic substances are toxic to microorganisms. Because of that, microorganisms cannot do the degradation process properly. Because of that, the BOD value can be decreased. Then dissolved oxygen, this is the chemical characteristics. So dissolved oxygen is required for the respiration of aerobic microorganisms as well as all other aerobic life forms. Okay, if you know the meaning of eutrophications, so in eutrophicated lakes, eutrophicated reservoirs, dissolved oxygen level is very low at night. And there are several stages in eutrophicated reservoirs, so you can learn about it in future lectures. Then we will see physical characteristics. So the most important physical characteristics is the solid content. Okay, So solids, then particle size distribution, because when we design the wastewater treatment plant, or when we operate the wastewater treatment plant, we should know the particle size distribution to install the proper functioning screeners or sievers. We should know the particle size distribution, and to design the sedimentation tank, hydrocyclones, also we need to know the particle size distribution. Then turbidity. In Sinhala, we can say it as Bora. Then color, odor, transmittance. Odor is happen due to the presence of some volatile matters. Then transmittance. Transmittance means, so what is conductivity? Okay, transmittance is due to the when the if there are metals in the water, what happen? Ions in the water, what happen? The conductivity can be changed. Then the temperature, density. Those are the important physical characteristics. So here you can see. So this glass has high turbidity, and this one does not has a low turbidity. Temperature. Why it is important? Temperature. So before we discharge the wastewater, before an industry discharge wastewater into a inland water body, they have to check the temperature, because it affects to the aquatic lives in the water. Let's see what is the temperature. Okay, here. Temperature discharge, it is shall not exceed 40 Celsius. Okay, and here BOD value, now you can see 30. COD value is 250. Oil and grease is 10. Total suspended solid is 50. Then you can find about sink. Okay, let's go to our previous slide. Okay, temperature. The temperature of water is very important parameter because of its effect on chemical reaction. So now you know temperature affects to the chemical reaction. If the temperature is increased, what happens? The chemical reactions, the rate of reaction is also increased. So if there are microorganisms, what happens? They will convert organic matter into simple end product consuming oxygen. So if the temperature is increased, the rate of reaction of the biodegradation is high. Therefore, at higher temperature, oxygen level in the water is very low. Okay, remember, at higher temperature, dissolved oxygen level is low. Except to that, there may be other reactions. Okay, the microorganisms degrade the organic matter biologically. So likewise, there may be some chemicals also, then the reaction rate is increases. Therefore, the minimum temperature, the maximum temperature of the wastewater which are discharged into the inland surface water should be maintained at 40 Celsius. So here now you can see the temperature effect to the chemical reaction, reaction rate, aquatic life, and suitability of the water for beneficial use. Increased temperature can cause a change in species of fish that can exist in the receiving water body. Okay. Change in wastewater temperature affect the settling rates, dissolved oxygen level, and biological action. Oxygen is less soluble in warm water than in cold water. What is the reason for that? So now you have to refer the Raoult's law and the Henry's law. So it is I will add about the Raoult's law and the Henry's law in our presentation. At the end of the presentation, you can check. According to the Raoult's law and Henry's law, oxygen is less soluble in warm water than cold water. So what happens then? The aquatic lives cannot respirate. They need oxygen to do their photosynthesis, and for the respiration they need oxygen. Oh, sorry, the for the photosynthesis they do not need oxygen. So if plant do the photosynthesis, they discharge oxygen into the water. Remember, when you learn about the eutrophication scenario, you will learn those things. Then sudden change in temperature can result in a high rate of mortality of aquatic life. Abnormally high temperature can foster the growth of undesirable water plants and water fungus. Remember, so it depends. So if we discharge high temperature water, it increase the growth of undesirable water plants and some type of water fungus, and that temperature may not suitable for some type of species, some type of fish species, some type of aquatic lives. So it depends. Then odors are caused by gases produced by the decomposition of organic matter or substances added to the wastewater. So if there are ammonia, if there are nitrogen compound, sulfate, so that odor can be generated. The most characteristic odor of stale or septic wastewater is that hydrogen sulfide. Odor is an indication of anoxic condition such as ammonia or H2S. Anoxic condition, that means there are little amount of oxygen. So if there are oxygen, what happen? Then ammonia will be converted into nitrate. Sulfur will be converted into sulfate. But if there are no dissolved oxygen, then they will be emitted as they will be converted into H2S. Odor produces psychological stress than to the harm to do the body. Offensive odors can cause poor appetite for food, nausea, and vomiting. Some odorous compounds are toxic at elevated concentration. Then turbidity. Okay. Turbidity is a measure of light transmitting properties of water. So in your practical class, you are going to measure the turbidity of the wastewater treatment sample. Okay. In coagulation and flocculation practical, you will measure the turbidity of the wastewater sample. So turbidity is the measure of light transmitting properties of the water. So here you can see in that picture, the NTU is taken as the unit for the turbidity measurement here. So this clear water sample, this is somewhat clear. It is 25 NTU. This is highly turbidity. There are many suspended solids, so it is 2000 NTU. So turbidity can be used to indicate the quality of the wastewater discharges and natural waters with respect to colloidal and residual suspended matter. So we'll check whether it is available in our tolerance limits. We'll check turbidity. No, it's not there. No, no, turbidity is not available. Let's see here. No, turbidity is not available. But in drinking water standards, it is available. Okay. So I think now you may be able to find about the standards for drinking water, because our chemical students have visited uh, water treatment plant in Ambatale last year. So now you are familiar with those things because you have learned during that field visit. Okay. The measurement of turbidity is based on comparison of the intensity of light scattered by a sample to the light penetrated by a reference suspension under same condition. So usually in turbidity meter we have to use a distilled water sample as the reference. Okay? The measurement of turbidity is based on the comparison of the intensity of light scattered by a sample to the light penetrated by a distilled water sample. Okay, colloidal matter will scatter or absorb light, thus prevent its transmission. So here there are colloidal matter, there are suspended solids. So if the size of the suspended solid is very small, then they are called as colloidal matters. So I will tell you in a future lecture what is the size of colloidal matter. So these are the very tiny colloidal matters. So what happened? The colloidal matter will scatter or absorb light, thus preventing the transmission. So here we will send an beam of light through the water containing beaker or one tube. Then the colloidal solids scatter and absorb. uh, Scatter and absorb. So if there are no colloidal solid, or if there are no suspended solids, all the 100% of light beam can transmit into the other end, but it is not possible due to the presence of colloidal solid. The result of turbidity measurement are reported as nephelometric turbidity units. So NTU is used as the units for the turbidity measurement, and there are other methods also. There is reasonable relationship between turbidity and total suspended solids. Okay, so here total suspended solid is a function of turbidity. Then that's why in our tolerance limits we can find only about the total suspended solid. So here uh, it is not mentioned the turbidity, but we can find the total suspended solids. Then solids. So in a wastewater sample, there are several types of solids: floating solids, settlable solids, which the solids which are very heavy, when the density is higher than the liquid, solids are settled at the bottom, so they are called as settlable solids. The floating solids are when the solid density is lower than the liquid, they are float on the surface of the water. And colloidal solids and suspended solids, they have same density and they are suspended. Okay, some of them do not have the same density, but colloidal means very small size and dissolved solids. So there are some symbols that we use, abbreviations. So TS is for total solid, TVS for total volatile solids, TFS total fixed solids, TSS total suspended solid, VSS volatile suspended solid, FSS for fixed suspended solid, TDS for total dissolved solid, VDS for volatile dissolved solid, and FDS for fixed dissolved solids. So my dear students, you have to remember all these things, but no need to worry. Those are very simple. When you do the practical, you will easily understand, and even in our next slide, you can remember those things. Those are available in the slide, and when you do the experiments, you will easily understand. So here, before I explain this, I'll show you this diagram. So here, when we do the practical, first we'll take a imhoff cone. No need to have a imhoff cone. We can take the water sample to a beaker. Okay. Then we put that water sample Either we can put that water sample to a petri dish, or we can keep it in a beaker also. Then we keep it in oven at 100 Celsius, and we kept it for 1 day, usually 8 hours or 1 day. Then we within 1 day we can see all the water has been evaporated, but only the solid contents are available. Then we can measure the total solids if we know the weight difference of the beaker before we put water into that, and after the evaporation process we can again measure the weight of the beaker. Then we can measure the total solids. So I'll show you here. Usually we take a petri dish or beaker, and we put known volume of wastewater, and we keep it in a oven. So this is the oven at 100 Celsius, usually for 24 hours. So actually, according to the, I think, standards, we have to keep it for 8 hours. But in university, it is not feasible to operate. It is not feasible to take readings within 8 hours. So we keep it for 24 hours. Then we can measure the total solid content. Then if we want to measure the set level solid, SS, here set level solids, we can put water to a Imhoff cone. Then at the bottom of the Imhoff cone, we can settle the set level solids. Then we can remove the water part. Then we can evaporate the moisture by keeping it in oven. Then we can measure the set level solid content. Then we can measure total suspended solids and total dissolved solids. Total suspended solids and total dissolved solids. For that, we have to get a water sample. Here, this is the water sample, non-volume. Then we take a filter paper and put it into a funnel, and we send the water sample through this filter paper. Then what happened? This is the filtrate, and this is the residues are remain on the filter paper. The residue remain on the filter paper are suspended. They are suspended solids. Okay, they are total suspended solids. The solids in the filtrate are total dissolved solids. So we can keep this filter paper in a muffle furnace. So this is a muffle furnace. We can keep this filter paper in a muffle furnace. That means we have to dry it in a muffle furnace. Then by measuring the weight difference, we can measure the volatile suspended solid and the fixed suspended solids. Usually the volatile suspended solids are evaporated during the drying process in the muffle furnace. So usually muffle furnace has to be operated in 550 Celsius. So the remain solids are fixed suspended solids. So if we take this filtrate, we can take this filtrate. Okay. There are total dissolved solid. So we can keep this filtrate in a muffle furnace. So when we put the filtrate into a muffle furnace, we have to keep them in a crucible, ceramic crucible. Then before we put the water, before we put the filtrate, it is better to clean and dry the crucible. Then we have to get the weight of the crucible. Then after we put filtrate, again we have to get the weight of the crucible. Then we keep it in the muffle furnace for drying at 550 Celsius for half an hour for 30 minutes. So after 30 minutes, we have to keep it in a desiccator. Then we measure the weight. Then we can get fixed dissolved solid content by taking the weight difference. Then we can find the volatile dissolved solid. Okay. So now here you can see there is very interesting. So here TSS, total suspended solid content, equal to VSS plus FSS. Total dissolved content is equal to VDS, volatile dissolved solid, plus fixed dissolved solid. Then if we know the fixed suspended solids and fixed dissolved solid, then we can find total fixed solids. So likewise, there are many, by using experimental method, we can find the total solid content. Okay, so this is the way of finding total solid content. Then in the first also I told you how to find the total solid. Then if we know one thing, if we know TFS, then if we know the TS, then we can find TVS. So don't worry, my dear students, you will learn this in practical class. So this is the reference textbook for water and wastewater treatment section, wastewater engineering, treatment and reuse. Usually 4th edition is available. So I have uploaded the soft copy of these textbooks into the Moodle, and this is written by Metcalf and Eddy. Those are the these authors of this book.